Hello, everyone. I'm Julie Matthews. I am a certified nutrition consultant with the BioIndividual Nutrition Institute and Nourishing Hope. And I am here with Dr. Bridget Briggs. And I would love to share a little bit more with you about our webinar today. We are going to be talking about methylation, bioindividual nutrition, and mental health. And I am Julie Matthews. I am a certified nutrition consultant and published researcher specializing in complex neurologic, digestive, and immune conditions, most specifically autism. I'm the author of Nourishing Hope for Autism and a co-author on a study showing the efficacy of diet and nutrition for autism, published in the peer-reviewed journal Nutrients. My approach is based on bioindividual nutrition, which stems from my 17 years of working with people with autism. And as you'll see as we get into the conversation today, it's really based on looking at the underlying factors that are involved in chronic conditions, digestive disorders, inflammation, all of these types of things, and how they influence the foods that we can eat and that we can't eat. And today we are going to be specifically talking about that in relationship to methylation and mental health as well. And so I have been using this model to help support practitioners that not only work with kids and autism, but all sorts of different types of chronic disease, including mental health disorders, as well as autoimmune conditions and all sorts of things. And so I also support parents as well. So I have training programs for practitioners and support programs for parents. And you can find out more about me at nourishinghope.com and bioindividualnutrition.com. Dr. Bridget Briggs, she is a medical doctor, board certified through the American Board of Family Medicine, specializing in integrative medicine. She has a private practice with locations in Temecula and Murrieta, California. She's certified through the Institute of Functional Medicine, the American Board of Integrative Holistic Medicine, and is a certified master in lipidemiology from the National Lipid Association. Dr. Briggs frequently lectures with other industry leaders on the clinical application of lifestyle medicine. A sought-after national speaker who talks on methylation are popular at most events. In fact, that's how I got the idea to do this, because she and I were both speaking at IMMH this year, and her talk was well attended. There were people trying to talk to her, ask her questions for over an hour afterwards. So that was what got me started. She's an expert in gut health, hormone balance, methylation, and children's health, and is well known in the inner circles of lifestyle medicine. This quest to teach came soon after her son was diagnosed at age 4 with brain cancer. During her son's horrific ordeal, she dove into research and discovered that many chronic conditions and diseases could be successfully treated using nutrition and lifestyle medicine. Dr. Briggs is the mother of 5 children and lives the life she preaches in Temecula, in California. She and her husband love doing all sorts of cool outdoor things, including camping and snowboarding and spending time at the beach in their gorgeous area of the country. And so welcome, Dr. Briggs. Thank you so much for joining us.
Thank you, Julie. It's good to be with you once again.
It's so great to have you here. And I know that people are going to be very excited to hear about this. By the way, they can find out more about you at drbriggs.com. And where else, any other resources that you want to share on where they can find more about you?
So we have a teaching site on epigeneticsrx.com. So this is complicated methylation and all the genetic SNPs. It's nothing I feel like any of us can be expected to learn in a 1-hour webinar. So right. We have pre-recorded webinars that people can join. They can look at some free ones. They can join a course on epigeneticsrx.com. So I think it's a great place to continue learning.
Wonderful. Absolutely. So as we're going to get ready to jump into our conversation today, I really wanted to go ahead. I have not too many, but just a few slides for people that are new to joining us. Welcome. In this actual session was originally created for our bioindividual nutrition practitioners. And so I wanted to open it up to the members that are going through our training program or who have gone through it before. And I just decided that this conversation and topic was so popular and so important among so many practitioners that I decided to go ahead and open it up to any practitioners or even some individuals out there that might want to join our conversation today. So I just wanted to welcome both the people that are joining us that I know, some of you from the program that are in here joining us. And for those of you who are new, welcome. And as I go through today, like as I said, I'll be sharing some slides and different information with you as we go. So let me go ahead and just do that to get us started. Just a reminder, this is for informational purposes only. And so today we're going to talk about methylation, and I won't go through all of this. I've got some great slides of Bridget's, but if those of you who know Nourishing Hope for Autism, this is a chart from my book, and we'll go through some of this later. I just wanted to introduce the topic, and then I'm going to let Bridget, I'm going to jump into some questions for Dr. Briggs. So the top part here is what I would call methylation, the part in blue, and she's going to, like I said, go through it in much more detail, but methylation is really the process of donating a methyl group to a substrate, and it's going to do something, and it does lots and lots of things. So there, we're going to talk about all the different things methylation can do. And then the reason I want to bring this up is that there's so many of these things that are connected. Methylation is connected to the folate cycle and a bunch of other cycles, and it also is important to then take us down into transsulfuration, which is the green part here, and then over to sulfation, which is the orange part. And that can affect many of the foods that we can tolerate and not tolerate. And it can also affect our mental health and a variety of other conditions and disorders. So we're going to jump into methylation and some of the different aspects of it. If for those of you who want to understand a little bit more about how this relates to bioindividual nutrition, for me, bioindividual nutrition is the practice of customizing diet and nutrition recommendations to the specific bioindividual and biochemical and underlying needs of the individual. And so we do that based on symptoms, their condition, health history, all sorts of things, including one of those things, what their genetics look like, what their SNPs look like, and what their biochemistry looks like. And so that's what I really do, and what I'm really known for is how to figure out which diet for which person. So that's the kind of context for our conversation today. I have this slide that's Dr. Briggs' slide, so I'm going to put this on the screen, and then I am going to just start with, maybe you can just start by tell us about just methylation to get us started here. And actually, before we even jump into this, I would love for people who are joining, one of the things I love about you as a doctor is you really understand the importance of diet and nutrition, particularly the use of special diets, which a lot of people don't do. Could you share for us a little bit of, firstly, how you discovered all of this and, how you brought this into your practice?
Sure. Thanks, Julie. So years ago, I went to one of my first IMMH lectures as just an attendee. And I was just diving back then, Julie, into genetic studies and genetic SNPs. And I actually started learning about specific nutrients from you, Julie. So doctors are not well educated or versed in nutrition whatsoever. And we have an understanding of histamine as a molecule, but we really have very little understanding about histaminergic foods or amine types of foods or foods that fall into the phenols. We all take chemistry, we take biochemistry, we learn, about these phenol molecules, but we don't really understand that they're part of food intake and the overlap between phenols, for example, and hormones and the configuration of estrogen and testosterone and how phenols can interact with some of these neurotransmitter degradation enzymes like your MAOA enzymes and how it would interfere with biogenic amines in the diet. So Julie, you were actually my first teacher.
Wow. Yeah. Wow. That's such an honor. Well,
you were the girl. You were the empowered nutritionist that brought my eyes to really truly understand the connection between diet and genetic SNPs and even nutrients.
Wow, what an honor. I didn't actually know that part of the story.
Yeah, you're the girl, Julie.
That's great. Well, that's That's wonderful. So, with all of that said, as we go through, maybe you can share some examples. I'm sure you will, because you're just loaded with examples. I would love to get into things as we go on, like the folate cycle and some of the SNPs that people are familiar with and maybe are new to people. But can you just give a kind of a high level overview of all of this and kind of how it all works together for people?
Sure. So I think we're all versed with this one little genetic SNP called MTHFR. And many of us all started with this as our first genetic SNP that we tested for. And many of us made some of the same mistakes that I made early on, thinking you could treat a single gene SNP at a time. So we would sit here and, integrate with giving patients methylfolate. And we didn't really understand that the introduction of this one little molecule was connected to these other gears or other metabolic pathways in the body. And so I remember early on when I gave a young teenager, some methylfolate, and she said it hurt her gut. And I'm like, oh no, methylfolate can't do that to you because it's just a folic acid. It's just a little B vitamin. And every time I tried to give it to her, I had upregulated her transsulfuration gear, and it was making more sulfur in her gut, and she was feeling bloated and gassy and gutty and itchy because I upregulated her histamine. So, as you make enough mistakes, you start to understand, hey, we turn on some other, pathways in the body, and how are they interconnected? So briefly, let's pretend like I'm talking to a patient, because we have to be low level when we're speaking to a patient. I always tell people, folic acid comes into the diet through, green leafy vegetables. And then our bodies use enzymes to convert it to the active form of methylfolate. And this little B vitamin acts like the first runner in a baton race. And we have to pass that little baton, which is a methyl group. And if I pass it over here to these two enzymes, I actually am able to activate homocysteine's conversion to methionine, which is a great way to reduce this rather neurotoxic and vascular toxic molecule homocysteine. But I actually successfully help in the conversion of the master methylator called SAMe. And really, all methylation begins with the appropriate donator of the methyl group called SAMe. And SAMe has the capacity, when the need of the body is for antioxidants, to donate the methyl group here by binding to the CBS enzyme, and it upregulates the transsulfuration gear, helping me to produce glutathione, the master antioxidant. And when the need of the body is to keep methylation going, SAMe will pass a methyl group by binding here to a cofactor spot on the MTHFR enzyme, and it actually turns MTHFR off. So when we have enough methyl groups, SAMe will turn this guy off, and when we need enough antioxidants, SAMe will bind CBS and turn this pathway on. So literally, methylation is like an on-off switch, turning reactions on and off according to the needs of the body. What most people don't realize is methylfolate is the only folic acid that crosses into the brain, and it produces BH2 into BH4, which is also coined the name biopterin. And biopterin is needed to produce both our monoamines like serotonin and our catecholamines like dopamine, noradrenaline, and epinephrine. So methylfolate is needed for both inhibitory and excitatory neurotransmitter production. And interestingly, that same methylfolate, when it increases biopterin, BH4 now binds the nitric oxide enzyme and converts arginine into nitric oxide. So if somebody has low biopterin because they have low methylfolate, they have nitric oxide issues, which is going to give you blood pressure problems, blood flow problems, cognitive, mental slowing, memory. And then this appropriate utilization of the citrulline, arginine, organic acids also keeps our Krebs cycle cycling. So we can basically see how this little red gear, the folate gear, is successful in keeping neurochemistry produced, nitric oxide produced, and the appropriate utilization of our molecules for the Krebs cycle, while folic acid has the capacity to turn reactions on and off when it flows through the methionine gear to simply passing that little baton. The methylfolate goes to the B12, producing methyl B12, passes it to homocysteine, now creating methionine, and ultimately the master methylator, SAMe, which again can turn on the transsulfuration gear. The reason it's important to know the flow of these gears, like in the student, the poor young girl that I said, if this gear is already fast and I activate too much of this gear, I can cause problems like histamine, itching, glutamate, insomnia, irritable bowel. Or if you're sulfur deficient, I can actually fix all those problems. So that's the brief summary.
Wow, it's great. All right, so let's go from there and just jump into some of these things one at a time here. And I brought up your other slide here on folate, because this is the one, as you said, a lot of people are most familiar with MTHFR. In fact, we already have an MTHFR question that was emailed into us, which I'll share in a moment. But I went ahead and I magnified this a little bit for people so they could see it a little bit more. Do you want to share a little bit more about folate, folic acid, methylfolate, whatever we want to know about this particular area?
Yes. Okay. So I always start up here. Here's our little folate again. And this little enzyme, dihydrofolic acid reductase, DHFR, acts on folate twice, once here and once here, to produce not only folinic acid, but this particular folinic acid is successful in producing the base pairs for DNA synthesis. So we always say how folic acid in pregnancy and embryogenesis is so critical, because we need not folic acid necessarily, but folinic acid and the 5,10-methylene for the production of our pyrimidines and our purines. So that's where DNA bases become important. Now imagine if you have a genetic SNP here in the MTHFR enzyme, and this kind of begins to stack up, Julie. Can you see how this might overactivate DNA synthesis, producing an excessive amount of purines and pyrimidines? Because we need to be able to convert that 5-methylene to the 5-methylfolate, which actually turns DNA synthesis off. So controlled synthesis and controlled turning off is dependent on healthy methylation.
It's very, very interesting. We always think more, more, more. And in this case, more is not always better.
No. And what's interesting is the number one drug, methotrexate, used in, for example, RA, rheumatoid arthritis. Remember, that's poisoning the DHFR enzyme. So if you poison this enzyme and you add back folic acid, can you understand that they're not converting the folate to the folinic acid? So it's a mistake made in rheumatology all the time. They don't give them leucovorin. Tetrahydrofolate is actually the prescription leucovorin. So just like people have MTHFR SNPs, we see people who have impaired fertility. They have impaired healing, that they can have this genetic SNP. And so by adding even leucovorin or folinic acid, you can correct even upstream genetic hiccups in folate.
Interesting. So, and let's talk a little bit about MTHFR, because there's different variants here. And you did a great job at IMMH kind of explaining to people how the level, the percentage of inhibition that we can get, whether we have the C677T or, whatever it might be. So can you share a little bit about those different SNPs and how that affects our ability to produce adequate MTHF?
Good. So, the most serious homozygote C677T, these individuals who have this genetic mutation, remember that they have only about a 30% functioning enzyme. So if you only have a 30% functioning enzyme, you may be fine chugging along throughout your day until all of a sudden one day when symptoms begin. So that could be pregnancy, right? This could be puberty because of hormones requiring that methyl group. So it's not like their enzyme doesn't work at all. It's just a 30% functioning enzyme. Now compare that to the MTHFR compound heterozygote. So we call them the CAs. I call us the California girls. Okay. The CAs, we actually function pretty much at a 50% capacity. So again, unless the need for methylation really accelerates, that may be fine, but you may begin to see symptoms when the need for methylation increases. But often in both those categories, the homozygote C and the compound heterozygote CA, you'll see little clinical manifestations. For example, cherry hemangiomas, geographical tongue, sometimes midline division like tethered frenulums or tethered tongue. So maybe mild developmental issues, but sometimes nothing dramatic unless, again, puberty or a birth control or like a wound or a cancer or something like that, or an autoimmune diagnosis where all of a sudden that 50% functioning enzyme becomes completely inefficient.
Mm. And if they're heterozygous for C677T, they've got about a 60% function. Is that correct?
Exactly.
But
And
people
now let's
I was going to say, people might not be aware that's very, very common. It's like 45% of the population has this, right?
and also think about the homozygote A129ACs, because looking at some of the research by Amy Yasko, it's really interesting because her research showed that the A129AC homozygote is a fast enzyme that fails to turn off. So actually, remember we talked about SAMe binds to the MTHFR enzyme, turning it off. So their folate gets drained into an excessive production of methylfolate till they eventually intracellularly, their polyglutamates of folate become depleted. So they still benefit from methylfolate oftentimes, but generally speaking, it's because the enzyme doesn't have the control switch to turn it off. So very different type of a genetic flaw.
Wow. Interesting. Okay. And then anything else here we want to share about B12? Because B12 is another important component of this, and then the MTR and the MTRR. Anything we want to say before we move on from here?
So let's just think about patients that are really methylfolate sensitive. And because of other genetic SNPs, what's interesting is methyl sensitive individuals sometimes can handle methylcobalamin. And if you remember the little methionine gear, the only thing you really need to stimulate the methionine gear to toggle back and forth between homocysteine and methionine is actually the methyl B12. If these two enzymes, which are the acceptor of the methyl group from folic acid, they convert cobalamin into methylcobalamin and homocysteine into methionine. If these enzymes are flawed or genetic SNPs, just remember they tend to be fast. So again, they suck up B12 in the patient, and the B12 patient becomes what we call a B12 craver, or sometimes a methyl B12 craver. So these guys love B12.
Interesting. So one of the questions that was asked already, because people were so excited that you were coming on, was the question around folate. She wanted to know, can you get enough methylfolate from your food? It turns out that natural foods like leafy greens and things have quite a bit of folate, whether it's a lot of it methylfolate, but other usable forms, more than, say, folic acid, which is going to be more in your processed added kinds of grains and things like that. So if we're talking your vegetables and all that good stuff, her question was, are people able to get enough? And are there some people that aren't able to get enough methylfolate from their food? And to share more on that.
So just like we see transcobalamin, genetic errors where people can't convert and absorb B12 and transport it, we do see some transportation errors even in folic acid. Some of those genes we didn't really cover in my lectures, but there are people that cannot transport and absorb folate as well as we like. You almost rarely see folic acid as a deficiency or folate serum levels that are deficient, almost never, right? So it's not generally we can't eat enough, it's how much methylation capacity. So we use MTHF as the primary methyl donor, but there are many other nutrients that we can eat, like choline or betaine, hydrochloric acid, things like that, that are also methyl donors, like magnesium citrate. And we can get those ingredients sometimes through supplementation and sometimes through food. But the real answer is, depending on the metabolic needs of the body, can the body, based on those genetic SNPs, produce enough methylation? And different stages of growth, development, and sickness and what have you determine that. For example, if we see little blonde towhead kids, they're blonde when they're little, and methylation matures as we age, especially around hormone production. So this is why our little towhead blonde kids get dark hair. That's through methylation. So methylation matures and changes as we age, as hormones develop. So we can produce more of those enzymes. Even if our enzymes are slow, we can make more of those enzymes based on the body's needs. But those enzymes also slow down as we age, which is why seniors may have a higher need for actual supplementation as compared to just eating those green leafy vegetables, because of the number of enzymes produced slows down as we age. So generally speaking, man was designed, even with this genetic SNP, to be able to eat the foods from the earth, but our environment, like plastics, is a potent demethylator. So we have to think about people who don't have genetic SNPs but may have excessive exposure to plastics may exhibit signs of demethylation.
Interesting. And then, of course, in autism, we have cerebral folate deficiency, where they're not able to transport it into the central nervous system,
Good.
where they might need more, right, than they could get from a food or foods. But yeah, that's great. Okay, that's wonderful. Let's see what else we've got here. We went through some of this, and then there, as you said, there's the different pathways, right? And you can do the pathway that involves choline. Do you want to say a little bit about that? Do you feel explain. So we talked about the folate portion, but there's also that conversion of homocysteine to methionine with that BHMT.
Right.
Do you want to say anything else?
So just to remember that this is only in the liver, because the liver's primary job is detoxification. So you can imagine methylation is a major process of detoxification in the liver. So this is not in the brain. This is only going to happen in the liver, but the liver has a secondary route that's capable of creating methyl groups. Even if the folic acid wasn't available, we have resources like choline and trimethylglycine and other glycine products, like we talked about magglycinate. But to remember that, for example, choline, which is in a part of the double phospholipid membrane, will be stolen from areas of brain and what have you when the folic acid is deficient, or the methylation from the folate and the B12 is deficient. The body is going to steal choline from other resources. So supplementation of choline kind of preserves like brain function in the biphospholipid membrane of cells.
Great. All right. And then I wanted to let's talk about CBS, because actually this is an area you taught me so much. You came up to me at one lecture that I did, and you were sharing all this really interesting information about what you do with sulfur. So I'd love to get into this. So for people, when we start with methylation and then we go down and try to create glutathione, she was mentioning when it go from homocysteine, instead of recycling back up, we can also go down. And that's what we're talking about here, is that little CBS enzyme right here that's going to take us down this pathway to make all the sulfur compounds and things. So share more on this, because this one I think can get a little tricky.
Okay, so this is a really fun pathway to wrap your head around, because you see so many individuals are very stressed in this pathway, meaning our CBSers, our patients that carry the CBS SNP, they tend to be fast. Okay. And they tend to actually have lower homocysteine levels. So they don't have high homocysteine. They tend to be very sensitive to B6. So when you give them too much B6, sometimes you'll see some of these amino acids like taurine will be very high in their serum amino acids, or in some of their organic acid GP3 studies and things like that. And what's interesting is cysteine, for example, is known to love itself. It wraps around and it hugs itself by making disulfide bonds. So people who tend to be dominant in this pathway, they tend to have curlier hair, thicker hair, higher foot arches. They tend to have things that are a little bit stiffer, so they're not very flexible. They tend to have more bursitis, plantar fasciitis, tinnitus in the ear. And that's because the higher the cysteine levels, again, the more the amino acids tend to kind of coil. And when this pathway is dominant, remember that they tend to have more gallbladder disease too, because taurine, when this taurine stacks, it's one of the taurine deoxycholic acid, or the bile acids that synthesize. So we see a lot of kind of gallbladder issues that surface here. It's critical in producing the master antioxidant glutathione, as cysteine tends to be one of those three amino acids for the production of glutathione. But in the rampant processing, there's often a high amount of ammonia that is released when this enzyme is active. So ammonia is one of those molecules that give people that brain fog when they talk about cobwebs in their brain, how they want to like pull the curtain aside. It's an easy blood test to measure along with the serum glutathione. But what's key is this pathway, there's a lot of neuropsych issues when people are dominant in the transsulfuration gear. And part of it is, number one, for every molecule of ammonia that is produced in the human body, it takes a molecule of biopterin. Remember our little BH4? So we need BH4 for brain production, neurotransmitters, and we need it for nitric oxide. But it will be consumed by a molecule of ammonia. So it's a 1-to-1 ratio. That's the whole gut-brain connection, right, Julie?
Mm-hmm.
And when cysteine is also produced from the homocysteine and the cystathionine pathway, we see a higher production of these sulfites. And many of these patients produce high hydrogen sulfide gas in the gut, so they have bloating and gas and heartburn and irritable bowel. They get SIBO all the time. We can measure this on some of our SIBO breath tests. So we measure methane and hydrogen ions. And because sulfites increase glutamate and histamine, they tend to be itchy, histaminergic. They tend to respond to like Pepcid AC and heartburn medicines because of all that histamine overstimulation of the heartburn. So this is that irritable bowel, bloating, gas, indigestion. I married one of these, Julie. That's why I just giggle over this pathway. As my husband and I, when we married, I was like Miss Broccoliaholic, every sulfur food because I'm sulfur deficient. And here he's this typical Scottish with the, you know, ruddy cheeks and the rosacea, all that sulfur histamine intolerance. And I'm like, I don't date sick guys. So if you want to date me, you have to eat like me. And this poor guy blew up his belly and he was always girty and gutty. And I told him he was going to die if he didn't eat all my sulfur foods. But
Oh, no.
we did the genetics and I'm like, wow, I guess I was wrong. And so I put him on manganese and molybdenum. And in 3 days, he's like, this is the best heartburn medicine I've ever taken in my life. My gut feels amazing.
Wow.
And that's because his little molybdenum here was activating the SUOX enzyme, converting his sulfites to his sulfates. And he was Mr. Tin Man. I mean, he couldn't even remotely bend over past his hip to touch his toes. He's just, you know, not at all flexible. And I'm this Gumby, touch the toes with my heels on, flat-footed, and totally two different subtypes. And, you know, in reality, when we began to clean out his sulfur pathways, his insomnia got better, his heartburn got better, his bloating. If he ate red meat, I knew when he ate sulfur. It was so funny. I would say, you smell like sulfur wine. I could smell sulfur in anything.
Wow.
So this has been a funny little one to kind of see how simple, from every H. pylori treatment he received to irritable bowel, it took 2 little minerals and your favorite low sulfite diet that you taught me, Julie. And so here I am, you know, preaching eat sulfur. I didn't even know I was saying that back then. I just thought you know, the glucosinolates, I thought everybody had to eat them. And then, you know, lo and behold, I looked back at his heritage. So this European gene, CBS, is commonly found in places of the world that there isn't a lot of sulfur veggies, you know, if you really think about it. So what are your Scottish and your Irish eat? Meat and potatoes. So it was adaptive for this enzyme to be accelerated and help to produce some of these other non-essential amino acids, but they're critical for bile acid production and glutathione. And some of the remnants of the sulfites needed more of those, you know, root vegetables to get those molybdenum and manganese trace minerals, which are just not abundant. And what's interesting is when you do a red blood cell manganese level on these people, and Molly, they're super deficient. I mean, they're so deficient because they have a higher need for those minerals than others. And ultimately, the good news is that with the accurate molybdenum and manganese, this healthy production of sulfates helps with the mucinous barrier of their gut. Sulfates add water to collagen and tendons, so their little tendinitis and their bursitis, their like lack of flexibility, the moisture comes back into those soft tissues, and then all of a sudden they're not so tight. And So sulfates are why we take glucosamine sulfate, chondroitin sulfate, because this process diminishes as we age. And so we get sulfate deficient, which also steals one of the important molecules that the liver needs for hormone detoxification and some of those monoamines like phenols that need to be detoxified. So this is a whole nother hormone lecture right there with the SOLT enzymes.
Yes. One of the things I teach my practitioners is exactly this, is that when you're looking at these issues like you just mentioned, whether it's CBS or SUOX, being careful of avoiding all the sulfites that are added, but also sometimes the sulfur foods. But this is where it gets tricky, because sulfur is also really important. As you said earlier, you know, we really do need that as well. So where is that fine balance? I'm always really nervous to tell people, don't do, do less sulfur, because I have seen too many people online, or people that come to me later and say, you know what, and this is a problem with sometimes the SNPs. I have X SNP, therefore I must do this. And that can sometimes get us into trouble, because as you know, it's much more complex than that. So and then so sometimes people have taken out the sulfur and they've gotten much worse, because we do need sulfur. It's very, very important to detox and all sorts of things. So how do you help patients with that fine balance of sometimes you need to be careful with the sulfur, but you don't want to do too low sulfur?
I think you're spot on, Julie, and you're right. I've made that mistake myself, like remove people on a complete low sulfur diet versus no sulfites, and then initially the symptoms get dramatically better and then they recur because of the sulfate deficiency. So I think the key is I do a short-term removal, like a 2-week removal, clean out the transsulfuration gear, and then do a slow add back in, but starting with the lower sulfite foods. So for example, kind of avoiding the additive ones like the sulfites in the wine and things like that, or like the dried fruits, like your dried apricots and things like that. And there are a couple of websites that go through like the picograms, the amount of sulfites that are found in specific foods. And it's quizzical. A lot of the sites are contradictory. They're not super accurate. So I truly believe, Julie, that the body craves what the body needs. And when your body, I have to always tell this story about this patient that I was doing her pap smear, and she asked me, little 21-year-old, she asked me, Dr. Briggs, why am I craving broccoli every night? I literally have to make my boyfriend go to Stater Brothers and buy me a head of broccoli every day. And I asked her, did you recently start a birth control pill? Because remember, estrogen is detoxified through sulfation. Right? Methylation, but also sulfation. So she goes, yeah, I did. And I'm like, well, because you dropped your estrogen, your sulfation enzyme slowed down. So your body's requiring you to eat more. So she knew. And my 2 little girls that are from my husband, they will take a green salad and all those dark leaves, I'll just pick them right out. And I just kind of let them flow with it. But when people are really out of balance, at least a 2-week cleaning and a slow reintroduction to find where is that sweet spot. But I do keep them on those 2 minerals, molybdenum and manganese.
Great. Wonderful. That's really helpful. All right. So let's let's see. What else do we want to say in here? Gosh. Oh, I know. I wanted to talk about MAOA and MAOB, because this has so much to do with amines and histamine. And then I want to jump into some with salicylates and other things like that when we get into sulfation. Actually, now that we're kind of down the transsulfuration pathway, we can let's take a little side detour into sulfation. Then we'll come back and we'll go into MAO and all that fun stuff. So this is one of the biggest areas that I find to make a huge difference for whether it's kids with autism, IBS, depression, and it's understanding of sulfation and how important and again, this is a process where we take sulfate and we, conjugate it with something, and we do hundreds of different things like the gut barrier, the brain barrier, detox. One of the things we detox or break down are these phenolic compounds, and they can be salicylates that are in plant foods like apples and berries and grapes and all those kinds of things. And they can be amines, phenolic amines, that are in things like bone broth and sauerkraut. Now, there's also histamines, but those are not processed through the sulfation. Those are processed through the MAOs and all that stuff. So we'll get to there. But anything you want to add, I don't know, maybe a clinical story or anything else you might want to add about sulfation and all of that fun phenol stuff?
Well, the thing that I want everybody to remember is, because a lot of our topic today is on impaired methylation, let's say we have a COMT, impaired methylation individual, where their catechol-methyltransferase enzyme is going to be so slow at methylation. Then the body is going to depend more on the sulfation pathway. Does that make sense? So our bodies try to compensate. If one enzyme is slow, it's going to put a bigger burden on this phenolsulfotransferase enzyme, which is going to be not only important in detoxifying those foods that you talked about, Julie, but also in estrone sulfate, which is a neutralized form of estrogen in phase 2 detoxification, as well as things like the phenol foods, the salicylate foods. And so what we're seeing is, for example, in our patients who have polycystic ovarian syndrome, we believe that this patient population has genetic SNPs in sulfation. And what happens is, when you measure their blood, their excessive amount of bisphenol A is 8 times higher than other individuals. So they have a sulfation genetic hiccup, and they're higher at risk for stacking BPA, which, remember, we talked earlier how BPA is such a demethylator. So it's going to strip methyl groups from these individuals. So we have to be able to look at creating a low phenol diet for these often COMT individuals, okay, because they have a lot more traffic going down the phenol enzymes, the hormones. And a low phenol diet, like taking the coffee away and taking some of those, teas away, and some of those spicy herbs and what have you that we use in cooking. We see this androgenic acne will surface like that when they're drinking coffees and high phenol teas, and we change their diet and put them on a low salicylate diet, and their acne gets better, and their really high DHEAS and estrone sulfate comes down. So I think that this enzyme, I don't feel like it's had a lot of attention, Julie, but you literally are probably one of the few practitioners, you and the Australians, that, because remember, people from Australia are fairly inbred. They marry each other, and so they seem to be very salicylate sensitive or phenol sensitive. Most of my understanding and research is coming through you and these Australians, which are phenomenal geneticists.
Yes, absolutely. Well, it's a good one, and it makes such a difference. I mean, I've seen kids with severe aggression I've seen it go away. I've seen just really amazing hyperactivity and irritability and mood and behavior things change radically from something that's really so simple as just changing from this food to that food. So yeah, that really came based on just clinical experience. Really, is what got me started in all of that. Let's talk more about COMT, which is, I'm rolling my eyes because I'm a COMT, homozygous COMT, one of my favorite SNPs. And then let's go into also MAOA and MAOB, because this can have an effect on those amines and histamines and other things like that. And it can have a big effect on mental health and anxiety and all of these being overly revved up and things. Any particular place that you want to start with talking a little bit more about that?
Yeah. So remember that our comp, Val-158s, the enzyme there's 2 variants, right? There's the wild type, which is a fast breakdown of your amines. So they're going to tend to have lower dopamine and lower catecholamine. So we describe them as your typical couch potato. They're like, oh, it's really hard to get off the couch and have motivation and getty up and go. So they tend to have lower dopamine. Okay. So we always call them the norm, but actually they're oftentimes dopamine cravers. So, they may have a higher tendency towards addictions, like trying to find the dopamine high. Okay. They do really well on anything that is dopaminergic. So for example, tyrosine, remember tyrosine is the amino acid that in the presence of the methylfolate is going to help to produce more dopamine. To remember that if you use a lot of 5-HTP, they both compete for the decarboxylase enzyme. So you can't put 5-HTP and tyrosine together. You have to separate them like 12 hours or whatever. But most of us are looking at the Val-158, remember? So their COMT enzyme is slow. You also have the heterozygote. So I'm the heterozygote, Julie, and your heterozygotes do tend to be more ADHD. Okay. So variant, right? So fast and slow. So the COMT Val-158s, they're the most interesting because they are described as wired and tired. And you can think, how could you be wired and tired at the same time? Because the brain is always in a mode of recuperating or responding to the imbalance. So the COMT enzyme is in the postsynaptic nerve. So after a fight or flight situation has occurred, so run, 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 saber-toothed tiger, got a deadline, the dopamine, the adrenaline is produced. Now, the MAO-B enzyme is in the presynaptic nerve. The MAOA is in the presynaptic nerve. But remember, the dopamine and what have you has been released. It's bound to the postsynaptic receptor. And now COMT is supposed to vacuum and remove that excitatory response. Well, guess what? It's slow. So these individuals, they're wired, they're amped. They tend to reside in a more stressed kind of a nature. They will describe themselves as tired because now the brain, the presynaptic nerve is being stingy. It doesn't want to produce and release a lot of the catecholamines, knowing full well that it's going to take you too long to digest it. They're also called warrior, meaning they are always like waiting for the ball to drop, right? So they feel this kind of low grade rev. But boy, if they get ticked off, if they're in a state of fight or flight, you do not want to be in their path because it's like a warrior, right? They're going to go in for the kill. Now, this makes them very type A, successful, high dopamine in the frontal lobe. They're some of your most brilliant individuals out there. That is not Bridget, that is Julie. Okay, so we need them. They tend to be, you know, I say the Spartacans in our culture. They saved the tribe, they saved the population, but they need to have time to come out of it, to rest, to ruminate. They like their own closets. They like to have their away time, their alone time. And as long as they can embrace that, the key is to remember that these guys get the adrenal burnout, right? So the catecholamines come from the adrenal gland. And if you keep burning that candle at both ends of the wick, these are the people that step into the fibromyalgia. They get the chronic fatigue. They suffer, especially during hormone changes. So women, we have the highest concentration of, you know, hormone phases in our life. So like at puberty, and have more exaggerated responses, postpartum depression, menopause is a real tough one on these individuals. And many times the balance of the COMT enzyme is hormone therapy or hormone balance. They're methyl sensitive. These are the people where I have a teacher. I just did this to her fairly recently. Can't believe she still loves me, but she is not only a COMT Val, she's an MAOB homozygote and an MTHFR. So she came to see me for stress, anxiety, the death of her husband. She has twin boys. She has 4 boys. She's a teacher. She's got a lot on her plate. She truly is superwoman. And I gave her a shot of methyl B12 and methylfolate in the clinic, and she literally hallucinated for about 15 minutes, amped, stressed, couldn't get on her little motorcycle and leave, so I had to stay with her. These people truly are sensitive to those excitatory catecholamines. And so COMTs, remember, you can help this enzyme by testosterone. So boys always do better than girls because they make more COMTs, girls make fewer. COMT is nurtured by magnesium. So these individuals do very well on magnesium sulfate, not glycinate, because glycinate is a methyl donor. Okay.
This is so interesting because every time I take a bath, I put Epsom salt in my bath. See, there's one of those intuitive things, I suppose.
Yes.
Fascinating.
They truly, truly love magnesium sulfate. The COMT enzyme, a lot of these people, because we talked about a lot of adrenaline, right? A lot of like go, go, go. So some of their fatigue can be nurtured with hydroxycobalamin and adenylcobalamin, but don't ever give them tons of methyl B12. It's really, sometimes they'll feel good 1 or 2 times, and then with chronic intake, they start feeling irritable, agitated, overstimulated, and they won't even always know it's one of the Bs that you're giving them. So just remember to proceed slowly with comps, nurture their parasympathetic nervous system. So they do very well with things that like panthenoic acid acetylators. You can help break down the catecholamines using niacin, like nicotinamide, riboside by Thorne and what have you. So they love to be demethylated. They feel calm, they feel mellow. Things like coffee and caffeine, those methyl donors, they may think they want them, but they get over revved, overstimulated. They can't turn off at night. So, you know, it's weird. They want coffee and I take it away and then they're mad and then they're happy. So, you know, they do very well on more of the calming foods. Interestingly enough about comps, because their methyl door is slow, they tend to be upregulated in the transsulfuration door. So they do tend to be a little bit sulfur sensitive, not like a CBSer, but they have to watch the amount of sulfur and the amount of histamines that are produced and the amount of glutamates that are produced in this pathway. So a more moderate version, they do well on Molly, they do well on manganese. So I call it my M&Ms. Magnesium, manganese, and Molly. Comps love my 3 M&Ms.
Interesting.
Yeah.
I'm making notes over here.
And my favorite brain food for them is Zymogen's Brain Sustain. No matter how neurotoxic they are, in about 10 days, 10 to 14 days, they feel mellow, calm, they deal with stress better. And it has 1,000 micrograms of methyl B12, but he put in like phosphatidylserine, acetyl L-carnitine in that thing, alpha lipoic acid, CoQ10. He put in so many nutrients. This is Dr. Perlmutter shake that brings down that excitotoxic. So I think they have the healthiest brain function on Brain Sustain. And what I love about Brain Sustain, it's almost a one-stop shop. So it's pricey, but comps end up coming in on 1 million ingredients, and they're not people that do well on tons of supplementation. So besides the magnesium and my little M&Ms, my 3 M&Ms, I try to be very cautious about overstimulating with too many nutrients from my comps.
And then let's bring in MAO, A and B now, and how this is going to affect, you know, what they can tolerate food-wise or other things.
Well, the most important thing about MAOAs is we always talk about, I think it's RS6323 is the common. It's actually a trifold gene. So it's kind of the way the 3 little genes come together, that we believe their serotonins are fluctuating. So remember, serotonin is an inhibitory neurotransmitter. It's going to be balancing. It's going to resolve stress. It's going to produce into melatonin. And so if your presynaptic MAOA enzyme keeps rapidly chewing up your serotonin and your noradrenaline, okay? So these are often some of your ADHD kids that they may have headaches because of low serotonin. They may not have the ability to calm down, resolve stress. They may be more explosive anger, but they may also have the lack of focus where they're not getting that norepi into epinephrine. So remember that our artificial adrenaline is like methylphenidate. That's Ritalin, right? It's a methyl donor. And so oftentimes these individuals end up on some of those ADD drugs or tons of headache medicines because we need that serotonin. So they're on every serotonergic drug, Imitrex, all of that, when they actually do really well with 5-HTP. So crosses the blood-brain barrier. Again, can't take it at the same time we give a bunch of tyrosine. But 5-HTP is almost like putting gas in your car. Okay, if your MAOA is irritable and keeps rapidly digesting your serotonin, then you have to put more gas in the tank. So 5-HTP is a great way to help your MAOAs. Remember, their key is serotonin concerns, but they also get noradrenaline problems. And if they have a noradrenaline problem, the problem is regulating that immune system, because norepi turns on and off the immune system. So 5-HTP is going to balance the serotonin. And then oftentimes being careful about excessive uses of stimulants in these individuals. Okay, so they tend to want to take Adderall, they want to take these drugs, and I'm like, I limit those. Okay. MAOB is a different beast. And I am cautious with our MAOBs, especially during hormone changes like puberty. We see so many of these teenagers coming in with the first break of bipolar type 2, and they're often misunderstood. They tend to have a nature of, they don't break down epinephrine. It's the biggest thing they don't break down. They don't break down histamine well, and all of those bioidogenic amines in the foods. Okay, so these guys, just the wrong diet is so destructive. They can't turn off at night. They can't relax. They're itchy. They're always throat clearing. The low amine diet is amazing. And they often know, Julie. They'll say, oh, I can't eat bananas. I can't eat avocados. I mean, they're a genotype that often knows some of those foods are very problematic. Well, so do the CVSers, a lot of times like, oh, I can't do sulfites in my wine and things like that. But the MAOBs respond incredible to the low amine diet. They do very, very well on things that blunt the epinephrine receptor, like propranolol or beta blockers, because once that adrenaline goes, I joke, they're the person, if you ever beep at them when they're standing by the car, they'll be plastered against the garage door, okay? Their startle reflex is ginormous, and their heart rates tend to be high. They're very type A, OCD. They like to control their stress because they don't have to worry about the adrenaline surge if they can control that stress. So it's a tough genotype. Again, they need a lot of parasympathetic. They need a lot of GABA. They are GABA-aholics. They crave every form of GABA, which makes them your alcoholics. It makes them the Xanax addicts, the benzodiazepines. So changing that diet gets them off their benzos. It helps them sleep. They're always like on some type of Ambien or, these like medicines that are associated with cognitive decline and memory problems. So watch your MAOBs. It's because the MAOA and the MAOB are on the X chromosome. If mom is homozygote, peg all of her boys. They're all going to be MAOBs. And let me tell you, MAOBs living together in a house is a lot of toxicity, right? It's like a lot of type A, kind of explosive. So when you explain that genotype to the boys, when they're irate and they're bullying and they're, temperamental, and you kind of teach them how they're going to react to stress, it helps them modulate and calm it down. They do really well with, again, magnesium sulfate. They do very well with phosphatidylserine. They do well with anything like L-mucuna, things that are GABAergic and supportive of dopamine, because their body starts being stingy with the dopamine because it doesn't want to make adrenaline.
This is, I'm laughing because you're describing me completely. And what's interesting I find is, well, it's amazing that I function considering, but I think that's the point here, is that I'm always surprised that the body, would it be nice if I had no SNPs at all? Sure. But it's interesting how whatever combination I have, they seem to support each other in some way. Sometimes they compound things negatively, but sometimes something slows down, but that's okay because it's not breaking down as much. And also we have to remember that we're not always expressing everything that we have. So I like to, just kind of put out that, not necessarily a warning, but, just because we're saying, if someone has this SNP doesn't mean that's expressing. And, we can't look at, if you have this one SNP, then always do this one thing, because we really have to look at them in the whole big picture. Because as you can see, it's an incredibly complex picture. So do you want to talk about that just a little bit about, how we, have to see this a little more broadly?
Sure. So I giggle because when I was at UCSD School of Medicine, I did a research project with Dr. Magulak. And the research project didn't involve herbs and supplements and genetic studies. It was a thesis on if a parent and a child had two different temperaments, and if we taught the parent about the child's temperament, could we make more functional adults? And so the study showed that let's say you have a type A MAOB mom. OCD, type A, very organized, controls her environment. And then you have this kind of methyl craver, ADHD child that's jumping all over the place, can't stay on focus. Harmony between that parent and the child are not going to come together real well. But let's say we have this kind of excessive catecholamine, dopamine kid that we say, okay, mom, we want you to integrate this child into some type of explosive sport like water polo, where this child's dumping all their adrenaline and their dopamine, rather than let's dunk your kid in a bath of mag sulfate. Hey, I mean, either way. But can we calm down neuroexcitotoxicity through either exercise, through calming baths, or removing foods? It's always been our history in nature that we've naturally crave the things that our brain wants. So, you know, the joke in my household is Nolan and I, my little son with the brain cancer, we are coffee cravers. Our best friend is coffee. You say coffee, I'm like, coffee? Because it's a methyl group, right? So we're creating exactly what our brain wants. And my husband, who shouldn't be drinking tons of sulfur, but you say red wine, he's like, red wine? Because he's craving GABA. So we're walking through life, and all of us are fairly good about medicating our neurochem. And these glimpse into our tendencies to get out of balance give us, you know, corrective, procedures to try to involve in our lives, and bringing in a nutrient can be corrective at times. But the patients, Julie, that are coming to see me, the wheels fell off the cart. Do you see what I'm saying? So I'm not talking you and Bridget. I'm not talking about some of our listeners that are highly functioning and we giggle and we recognize a little tendency that we all have. But many of the people that are coming to see us, it's because something went wrong. Balance was lost. And so knowing that there are, you know, biochemical reasons that they've gotten off balance, and ingredients that we can do and diets that we can implement, it's just empowering.
Absolutely. Absolutely. That's great. I love that. Thank you for sharing that. So let's go ahead. I just want to sort of wrap this up a little bit in terms of bringing it all together here. Kind of, you know, part of the reason that we're here is to just kind of explain that when we look at whether it's methylation and various SNPs, or, you know, how things are functioning in these biochemical pathways, what can we do? And as you said, there's a lot that we can do. We can work on our lifestyle with exercise, whether it's baths, whether it's calming things, whether it's supplementation support, and also diets. And so that's what I do, and that's what I try to help my both my clients and the practitioners that take my BioIndividual nutrition training, is to figure out what people need for what. And so just to kind of bring the conversation around. So we talked about if we get down to if we have a breakdown in the orange area down in sulfation, then we might need to be careful of phenols, or we might need to be careful of various types of phenols, whether they're salicylates or biogenic amines. Also, we didn't really talk today, but oxalates, for example, can be transported into the cell on the sulfate transporter when we don't have enough sulfate. So when people have a challenge with whether it's methylation, transsulfuration, anything that's any of those enzymes and genes that are going to get us all the way down to that orange area, we want to be careful of those types of things, or at least be aware, have a sense of how these might affect someone. So I might do a low salicylate diet, or a low amine diet, or both. I might need to do a low glutamate diet or a low oxalate diet. We might have to look at a low sulfur diet or a low sulfite diet. When we get we don't have enough glutathione, we don't have enough sulfate, we don't have enough gut barrier, and so we might tend to be more having food intolerances, food sensitivities. We look at various ways that we might have more histamine reactions and things like that. We might need to be careful, not only histamine foods, but other types of foods that might kick up inflammation, or other food allergen reactions and things like that. And so that's really kind of the bigger picture here of what we're talking about. So I'm hoping today you got some valuable information, the people that are on the line with us. If you're in our program already, we're going to have this interview in that back area so you can listen to it again. And I'm also going to be putting together, or I'm going to be uploading handout for you in that membership area that goes into this chart and lays out things we want to bring in, things we want to be careful not to have in our diet, so we can figure out, what do we need more of and what do we need to avoid or have less of. So if you're in the program, those will be there. And then for those people that want to, we are going to do a fall enrollment this fall. So email me if you want. I think I have my email on here somewhere, info@bioindividualnutrition.com, and let me know that you're interested in learning more about the program. We can, get you whatever information that you need for that. So that is, will take us to the question and answer portion. So I'm going to stop sharing my screen right now, and then we'll go to that. All right. One of the questions that a bunch of our practitioners had asked, and they wanted me to ask you, which is, how do you know if you're an over-methylator or an under-methylator? Can you use just SNPs to figure that? Do you use more metabolites that are down that pathway? How do you figure it? Can you simplify it to over and under-methylators, or is that too simplistic?
I don't use that terminology, Julie. I use the terminology of methyl craver or methyl sensitive.
Okay.
So if you're a methyl craver and you take methyl groups, you feel good on them. You feel focused. The joke in my clinic is they'll say, Dr. Briggs, you didn't take your B vitamins today, did you? Because I'm all over the place. And that's no different than when people take a cup of coffee and they're like, oh, I got to have my coffee today because you're craving methyl groups. You just don't understand that that's what you're craving. It's when you take your Ritalin drug, methylphenidate, when people are like, oh, I'm not focused today if I don't take my methylphenidate. Well, what you're saying is I'm not methylating. So I don't give methylphenidate. I try to give methyl groups. So methyl groups are robust. Remember SAMe, methyl B12, methylfolate, trimethylglycine, choline, glycinate. Those are all different forms of methyl donors. So if you're a methyl craver, I always say that you can always look at the color of your urine, because when you're methylating and you have enough, you spill the remnant out of the urine. And so it's bright neon yellow, meaning you had too much and you let it go. Hooray. When you're undermethylated and you're taking methyl groups, your urine is clear. And usually something's brewing. So for example, I always know about once a year I get laryngitis, and I can almost always predict when it's coming because I take the same methyl groups every day. I take 3 methyl B complexes in the morning and 3 at night, and my urine's yellow and it runs out by 3 o'clock. And so I take an evening dose. And right about the time I'm going to get a cough or a cold or a laryngitis for about 2 days, I have clear urine, even though I'm on the same Bs. I'm like, oh, something's coming. And if I take more methyl Bs, it doesn't rescue me. Does that make sense? It's oh, something's coming. So my immune cells during the cell division are ramping up to probably go make my, immunoglobulins that are in my lymph nodes go and kill whatever. So I never get really sick. Everybody jokes like Bridget gets hoarse for 2 days and then she's back to normal again. So, it does boost your immune system, but you've been exposed to something. Now, if you take more methyl Bs and you're trying to get more yellow urine, guess what? I mean, once you have enough, you're just going to keep peeing it out. So more isn't better.
Right.
Now, methyl sensitive individuals, this is like my sister. I give her a methyl B12 and she's up till 2 a.m. cleaning the house. And she's like, yeah, I mean, that's great. I'm cleaning the house, but that's way too much energy, Bridget. So that's just too much methyl. Right? Does that make sense? So
yes.
Overstimulated. And sometimes it's subliminal where, Dr. Loudon, my son's neurosurgeon, I put him on brain sustain for the first couple of weeks. I actually gave him a methyl B12 shot. We didn't even know his genotype yet. And the first time he's like, oh my gosh, my neuropathy is better, Bridget. I feel like $1 million. Week 2, can I have another shot? I gave him another shot. Week 3 came in. He's like, I'm like, you're fretting. What's up? He's like, I don't know. And I'm just like, really worried. I'm really stressed. I'm like, oh, he's overmethylated. So he was like revved. He was overstimulated. He was pulsating in that typical COMT, MAOB compound. He's got both of those genetic SNPs, so too much adrenaline. So I gave him nice, and he's like, that feels better. So you have to recognize that excitotoxicity, kind of that rev, that you may just feel like you're having a bad day, but that rev is too much adrenaline.
Very good. All right. Let's see. Someone wanted to know which company you use for manganese and molybdenum.
So I built a product and I called it Sulfite Double X. You could build it yourself. It's nothing magical. It's sold by epigeneticsrx.com. So many people just go to their website. They mail out the moly and the manganese. I've just come to be very careful by not telling patients to just go over the counter and do it themselves, because I had one lady that took 80 milligrams of manganese and ended up in the ER with, manganese toxicity.
Oh, no.
She's a fibro patient. She felt amazing. So she felt like, hey, let's take more and more and more. No, you can get toxic on manganese. And moly is super clean. You can't get toxic on moly, but I just put it together. It's 600 micrograms of moly twice a day, and manganese is 10 milligrams twice a day. I just put them in the same supplement.
Okay, great. What did you say about high taurine again? What does that relate to?
So people that are over-revved, like their transsulfuration pathway is over-stimulated or overproduced, they tend to have, for example, on the Nutri-Eval by Genova, taurine levels over 1,500. So that is just an evidence that pathway, that sulfite pathway, is really, really high. Mm-hmm. And they tend to have excitotoxicity, insomnia, the joint issues, the bursitis issues. And then the sulfite double X will help to reduce that. But you have to reduce some of those high sulfur foods, like especially the meat, like the red meat and things like that. So I'd rather take out the animal meat in terms of sulfur amino acids rather than taking out all my veggies if I could.
Mm-hmm. Okay. So let's see. Do you have notes or a book on the subject? Your lecture is wonderful and the slides incredible.
So remember, we have those webinars. I do have 4 chapters of a book, but the book is not complete. And with my 5 kids, Julie, I don't even know how you wrote a book.
I understand.
It's insane. Oh my gosh. Anyway, so one day I promise it's coming.
Great.
But we did find that live things like this and listening to people was a little bit easier than reading a book. It's really boring. This is not super exciting stuff. Stories help. So the webinars that are on epigeneticsrx.com, they're 20-minute webinars, a little short. We went through each gear. We give 3 case examples on every webinar. I want to say the core class has 16 webinars. So super doable. Put it in the car, drive in a car, really easy. And then there's day-long seminars. So this is complicated. I wish somebody wrote a book. I don't know that it's going to be Bridget, but somebody needs to write a book.
I hear you. And if people are interested in my book, that slide that I put in there is in the book, and I have, I think, maybe 30 pages on all of this methylation transsulfuration stuff, and that's Nourishing Hope for Autism. It's people buy it that don't have kids with autism. I find the pathways and the info is the same, and actually that's one of the things I discovered working with autism is that this doesn't help just people with autism. It helps people with all of the types of things. All of these SNPs and things that we see in autism, we see in anxiety, depression, heart disease, all these things that Dr. Briggs has been talking about. And so a lot of these principles and the diet principles can be used in various conditions as well. So if that's helpful, that exists, it's out there. So let's see, what else do we have? How important do you think diet is to this whole picture?
It's everything. Everything. People underestimate the power of nutrients. And doing this, I've been in this valley for 19 years. It wasn't until I really embraced your diets, Julie, literally. Diets are the one thing that it's not a pill, it's not a supplement, and it empowers the patient. Patients do not want to swallow a million supplements. They don't. They're willing to a point, but they feel control when you give them, this is your diet. Sometimes they feel sad and deprived, but they usually wrap their heads around it, and then they modify it according to the nutrients that they need and their body craves. So there is literally a diet for every genotype, except for mine, the ADD diet. My ADD diet is drink coffee till, all nights of the day. But hey, the research on coffee right now is very enlightening.
Yes, unless you're me, in which case I cannot handle a drop of it. I was that person where I loved coffee, and I'm Italian, and I just love coffee, and I just can't do it. And I was very sad for a long time. My husband, he's the opposite. He can drink all the coffee, and I have to smell coffee. But now I am so happy. Like years later, I I don't even want it. It's like you said, it's like once you know the pain that it causes you, it's easy to just to let go of it. So,
Yes. I
so
would be very sad if that was my genotype, but right now my genotype is just like, where's the coffee? I love it.
yes, yes, yes. So, okay, great. Well, thank you for sharing that, because I think it's really, I think sometimes we all want the easy fix or the simple thing, but sometimes diet is really important. And like you said, when you start doing it and you start to feel better, then you don't mind doing it. It's something that you look forward to. You don't even miss the things that you used to love because you just feel so much better. So I definitely support that idea of, give the diet a shot. If you're a practitioner and you aren't versed on all this, come join us in the Institute and take some training and just learn what I learned in 10 years and just download it all at once. And you don't have to learn by all the trial and error that Dr. Briggs and I have done over the years. So can you repeat what you said on B12 cravers? I have pernicious anemia, MTHFR C677T, and FOLHI. I'm a runner athlete who seems to deplete, and cells are still deficient after frequent shots.
So Jenny, what's really interesting about B12 orally, the research shows us that if you were to take 1,000 micrograms of B12, you could actually only absorb 5 to 7% of it. So many individuals, if you look at B12 in the blood and they're not even taking B12, it'll be over 1900 or 1999. And they'll ask me, how is my B12 this high? I don't even take it, because they have a transportation error, right? Either a transcobalamin transportation error or what have you. So first of all, humans need intrinsic factor to absorb B12. So you can have an intrinsic factor deficiency, you can have pernicious anemia, but sublingual is always a great way. But ultimately, I would consider you, Jenny, more than likely an individual who needs an IM injectable. So you can saturate your cells. You can look at your methylmalonic acid. There's some other markers in the Nutri-Eval by Genova that will tell you when you're finally getting those methyl groups in your body. But I would go by injection. It's just going to be really hard for you to do it orally.
Okay. Someone says, so COMT heterozygous, do you want to demethylate? And then there's an add-on, COMT plus plus or COMT plus minus do better on brain sustain?
So the COMT heterozygotes, they still tend to receive methyl groups very well. So I'm not sure if you're always going to call them a methyl craver, but they receive methyl groups without getting overstimulated. The heterozygote COMTs tend to be more ADD, right? So they're not excitotoxic. They're more like hyper-focused, right? So they still have a lot of the dopamine, but a lot of times an imbalance in the full balance of the catecholamines, because their methyl transfer enzyme, COMT, is going to transfer the methyl group from dopamine to noradrenaline and noradrenaline to epinephrine. So usually dopamine is the highest, norepi's a little bit lower, and then epinephrine's lower. So they're like your energy cravers, like, where's my stimulant? Okay. Meaning they like methyl groups like coffee, like Bridget. Okay. Now, if they're also an MAOB, be careful, right? So you have to look at genes together.
Yes.
But your COMT homozygotes, they're methyl sensitive, right? So they do very well on brain sustain because they tend to be excitotoxic, right? So a brain that's like way too active. So brain sustain is very neurologically calming. Your heteros may do better with focus, but they don't necessarily, they're not excitotoxic. They're more like focus is the problem, not anger, explosion, coping, overstimulation, sleep. They're usually, honestly, they're your doctors. I am telling you now, there's a reason that every doctor in the hospital has a cup of coffee 5 times a day. All the doctors that are my patients, they come to get their Adderall. It's really sad. And they're all a bit of ADD, literally. That's why you can't get a doctor to focus. So they don't tend to be your COMTs. They don't. You can't stay awake multiple nights in residency as a COMT. Do you understand? I mean, they crash. They need their rest. So in residency, I think I didn't sleep every other day. I was OB-GYN, so we didn't even have a bed. So, I usually categorize people. You're really, type A, highly successful brainiac. They're either an MAOB or a COMT.
Or both, like me.
Yeah. That's a tough genotype. It's really tough.
It's tough.
You have to be that smart, Julie.
Well, I don't know if that's what's tough about it.
It's true. It's true.
I'm going to come see you, Bridget. Okay, Dr. Briggs, I saw you at IMMH. I enjoyed it. You mentioned a genetic test you use. Wondering what test do you use? Can a patient order it themselves, or does a doctor need to do it?
So I order Myriad Genomics, which is right here in my backyard, believe it or not. How crazy is that? They did create a genetic SNP profile. It's a 2-page, I don't even know, like over 100 genetic SNPs, but they contain all of the methylation ones. So MAOA, MAOB, MTR, all of them, and more. So like some of the folic acid SNPs, FUT2, transportation. It's called Myriad Genomics. Any practitioner can order it for a patient. It's $250, but they have a geneticist that meets with the patient and helps you with the interpretation, because it's pretty deep. It's really deep. Now, a lot of people have done their 23andMe's, but there's about a 45% error rate in 23andMe. So you got to be real careful.
Yikes, that's not good.
Yeah.
Tell me that, say it slowly, say what the name of the company is.
Myriad Genomics.
Myriad. Got it.
So the Epigenics RX website can get you all connected as a practitioner, so you can order directly through them, or you just call Myriad Genomics and set up your own account. Great. The geneticist comes from the Epigenetics RX group. So they charge the patient, I want to say $49 for a sit-down Skype meeting like we're having right now to go through all their genetics.
Great.
Pretty good deal.
That's awesome. All right. Let's see. I know we're kind of running over on time. I have like tons of people with questions, and I have people chatting more questions. People are saying the information is priceless. Someone mentioned Dr. Walsh's great book on nutrient power. Let's see. Okay, so let's go and let's see if we can add a few more questions, and then we'll wrap it up. I'll do my best to get to as many as we can here. An explanation for nighttime sweating every time after an Epsom salt bath.
So I always think if you're still sweating and you just took an Epsom salt bath, which is going to break down all those catecholamines, sweating would be more low serotonin. So I would work more on 5-HTP.
Interesting. All right. Do you have to be a physician to study with epigenetics RX, or
all kinds of practitioners?
Great, and lay people too, right? Isn't there a consumer? Yeah.
Yes.
Great. Okay. Is your program code? Okay. We got that. Okay, can you say again, what was the chemical hormone that's causing the startle reflex in the MAOB SNPs?
Inability to break down adrenaline.
Okay.
Their heart rates are revved and they have higher blood pressure. It's adrenaline.
Got it. And the Pearl Mutter shake was brain sustain, right?
Mm-hmm.
All right. Please comment on the Walsh approach for treating undermethylators and overmethylators, i.e. when whole blood histamine measurement indicates undermethylation, then according to Walsh, one should need to limit folate and not ever be given folate supplements, as folate destroys methyl within the nucleus of the cell. What do you think?
I'm not sure that I understood that question. So whole blood histamine measurement indicates undermethylation. Then according to Dr. Walsh, one would need to limit folate. All I know is if you look at your Nutri-Eval, the histidine pathway associated with, whether you're converting your polyglutamates to methylfolate gives you an idea if the polyglutamates are not becoming sufficiently turned into the methylfolate. But guys, I don't really do those kind of studies. I use doctors' data, global methylation panel, to look at the SAM-SAW ratio. So ultimately, if you're appropriately methylated, you make the end product, the global methylator, called SAMe. So you don't need to keep adding methylfolate. You can affect the end product, which is SAMe, by just simply saturating a door that can't convert to SAMe. So if you want to know how much to give, get the global methylation panel test to assess your sweet spot.
Great. I'm a heterozygous for the C677T MTHFR mutation, and my orthomolecular psychiatrist has me taking methylated vitamins. Do I need to take these supplements for the rest of my life?
It depends if you have methylation requirements or needs. So just because you're a heterozygote doesn't mean you need methylfolate. It's, does it have a balancing impact? So for example, if you can't tell a story, if I take methyl Bs, I don't get geographical tongue. I have a really bad geographical tongue. If I go to the dentist and he does my gum checks, he's like, oh, you started flossing your teeth. No, I'm taking my B vitamins, but he doesn't know that. So it's not like, oh, my brain feels like this. Sometimes it's looking at things like cell division. How quickly do your nails grow, your hair grow? So if you're like, my hair and my nails don't grow, methylate. If all of a sudden cell division is normal, then you needed it. So you should be able to tell a story. There should be something that you noticed A, B, C changed. If not, I don't think you need it.
Great. And then someone says, can you take GSH instead of MO?
So they're saying, can you take glutathione?
I think they're saying, can you take
Molly?
Yeah.
Okay, glutathione is very expensive. Liposomal glutathione, extremely expensive, and you should be able to produce your own glutathione on an as-is-need basis. So between the two, Molly's pennies. Molly still isn't going to get you to glutathione exactly. So remember NAC. N-acetylcysteine is really the rate-limiting step in the production of glutathione, which is the master antioxidant. So again, also surfaces on NutriEval. Also glutathione levels can be measured by LabCorp. So if you really needed it, you should show me that it was low and that you had to take it to replace the glutathione to be normal in the blood. I never give glutathione. I just methylate and people produce glutathione.
There's an interesting study in autism showing that when they gave folinic acid and got that whole thing going, then they were able to make much more glutathione. So yeah, great.
Simple blood test.
Yes. I think that's it. I think we're in good shape now. Wow, that was so great. Thank you so much. I knew this was going to be amazing, of course.
Thank you, Julie, for having me. Thanks,
Absolutely.
everybody.
Yes. So let people know one more time, drbriggs.com and epigeneticsrx.com is where they can reach you. And you have a practice in California, Southern California, for people that want to come see you. And lots of people are saying thank you and, you know, really excited about the information. So I just want to say thank you again.
Thank you, Julie, for having me.
All right. Thanks, everyone.
Thank you. Bye, guys.
Bye. Thanks for joining us. And again, I'm Julie Matthews, a certified nutrition consultant with BioIndividual Nutrition and Nourishing Hope. And if there's anything I can do for you, feel free to reach out to us at info@bioindividualnutrition.com. All right. Thanks, everyone. Have a great day. Bye.
Bye.